Hi, and welcome to today's episode of the Oxford Centre for Personalised Medicine Flash Interview Series, where we discuss aspects of personalised medicine with people involved in it in one way or another. Our guest today is Dr. Julia Foreman, the Decipher Project Manager. Hi, Julia. So it's great to speak to you. Could you begin by telling us a bit more about you and your story so far?
Hi, yes, of course. So my background's actually in research. So my early career, my PhD and early postdocs was in molecular biology and genetics, but it was actually in plant developmental biology. So I moved to managing biology projects with a strong genetic element around 10 years ago. And I refocused to working in human genetics about 7 years ago. So I've been managing the Decipher project for just over 5 years.
Brilliant. So the Decipher Project then, what exactly is it? Why did it come about? And what did you aim to achieve with the project?
So Decipher is a website and database. It shares rare disease patient data and provides interpretation interfaces. It's a record-based system and it has both genotype and phenotype information. And the data is deposited by academic centres worldwide. So the main aim of Decipher is to drive diagnosis and discovery in rare disease. So Decipher was established in 2004, and this was when the first CMA results were coming out. So what the clinicians were finding is there were no sort of really good tools to see which genes overlap the copy number variant found in their patients. So it was obviously very difficult to interpret that result. So the first Decipher, the first version of Decipher, actually what it did is you could put your genomic coordinates in and you can see which genes overlapped your CMV. And obviously at that time, many of the CMVs that were found had not been seen previously. So what you needed to do then was share the information to find similar patients and then hopefully find a diagnosis for your patient. Since that time, so obviously Decipher has been running, 17 years? The platform has moved on, it's been developed. So around 8 years ago, we started supporting deposition interpretation of sequence variants. And last year, we actually moved to support all types of genetic variation. So that includes aneuploidy, UPD, inversions, insertions, and short tandem repeats. So one of the key things about Decipher is it brings together the variant and the phenotype information. So it allows you to easily determine if you've got a good phenotype match. So it doesn't actually give you the diagnosis of the patient, it tells you those granular phenotypes. So this not only allows diagnosis, but it allows discovery. So it allows you to find new gene disease associations. For example, a new phenotypes associated with a disease. One of the other main aims of Decipher is to integrate new research data sets and tools into the platform so that we can quickly translate that information into the clinical arena for patient benefit.
Brilliant. So what have been some of the biggest successes of the project so far?
So I think one of the big successes, obviously, we've been supporting genomic community for over 17 years now. We now have more than 40,000 open access patient records available to anyone that visits the website. It is a live website, so all of those records are updated immediately when one of the depositing centres updates a record. We also support consortium sharing, so that's sharing between specific centres. For example, we support the NHS Consortium, so that allows the sharing between NHS centres of the anonymised patient records before there's explicit patient consent for that open sharing. And we currently share more than 65,000 patient records in that way. Decipher has had a huge impact on rare disease diagnosis and discovery. And we know that because there's been more than 2,600 publications that have cited Decipher since its inception. Decipher also supports genomic medicine across the world. So that includes countries with developing genomic services. I think one of the reasons that Decipher has been so successful is that we have an oversight of the whole process. And so when I say that we speak to the researchers who are bringing out the new research data sets and the tools, the developers working on Decipher are involved in those discussions. They know the data sets well, they understand why they're being integrated. And our clinical lead, Helen Firth, is a consultant clinical geneticist and she sees patients and she uses Decipher in the clinic. So we have the oversight from the researchers producing the data all the way to the clinic.
So I guess then on the flip side, what have been some of the biggest challenges and the hurdles that you've faced since Decipher's conception?
So I would say one of the challenges has been maintaining a balance between sharing the rare disease patient data and balancing that with patient privacy. So obviously Decipher's used worldwide. There's regulations differ in different countries. And it's also quite a changing landscape. So people's ideas and perceptions and those regulations are not set in stone, they do change. So Decipher takes a proportionate data sharing approach. We only share the data that's required. So we share a small amount of variant data, so those variants that are most likely to be responsible for the clinical phenotype seen in your patient, along with that granular phenotype information. We have maximum reach. The data is shared completely openly. So to allow that, Decipher is designed very carefully to protect patient privacy. We very carefully consider what data is deposited and how we display that data. For example, we do accept some quantitative data, but we share that in an aggregated format to protect the privacy of the patient. If people do require more granular information than that is available on Decipher, we do allow a mechanism for them to contact the depositing clinician. So in that way, they have access to that more informative information. To allow us to do this, Decipher does have research ethics approval, and we also have an advisory board. And we have representatives not from clinicians, just from clinicians and researchers worldwide, but also representatives from ethics, law and patient representatives. So the CEO of Unique and also the Director of Policy from Genetic Alliance are on that advisory board. So obviously one of the challenges for genomic medicine is personalised medicine. So Decipher is very well set up for this. We have a very strong connection between variants and phenotypes, and with Decipher, you can help identify the different subtypes of patients with a particular disease. In that way, you can tailor the treatment. We also allow the grouping of variants. So in the future, you might have a variant, a pathogenic variant that's causing the phenotype. There's also a modifier and maybe a polygenic risk score. It may be the combination of those three that's actually causing the phenotype seen in your patient. And within Decipher, we already allow the grouping of variants. And in the future, we will also support polygenic risk scores, and you'll be able to group that together.
Great. So I guess that brings me on to my last question then about what the future is for the Decipher project and where you're going in the next decade or so.
So we've got lots of exciting developments planned for Decipher. So one of the things that we want to support more is the interpretation of variants in the non-coding genome. So this includes things like regulatory elements. We're planning, obviously, incorporating any good new data sets that come out. So next year, NOMAD version 4 will be released. So that big population data set will be even larger, and will also include a greater ethnic diversity. We're looking to include functional data. So if there's functional data available, we'll be able to present that in Decipher so it's easily available in the clinic. And one of the things that we're imminently going to release is the visualization of your patient's variant on the AlphaFold predicted protein structures. So within Decipher, there's a protein browser. And on that protein browser, you can click, you can see a 3D structure, and you can see exactly where your patient's variant's located. And those AlphaFold predicted protein structures were made openly available at the end of July, and very, very soon they'll be available in Decipher. So it'll be available to clinicians within the clinic.
Lovely. So I guess it's just watch this space then for Decipher going forward. Thanks a lot for your time. Is there anything else you wanted to add before we finish?
No, just thank you very much for having me and letting me speak about Decipher.
Lovely. Thanks very much.